Hi, my name is Johann Kruschwitz. I'm a postdoctoral researcher at the Department of Psychiatry and Psychotherapy at the Division of Mind and Brain Research at the Charité Hospital in Berlin. And today I want to give you some ideas about graph theoretical modeling of brain connectivity. And specifically, I want to focus on some concepts and the workflow of brain connectivity analysis using graph theory. First of all, I want to acknowledge Daniel Bassett and Alex Vernito because they gave me permissions to show some slides of their previous talks during this little introduction into graph theoretical modeling of brain connectivity. So the human brain is really complex over multiple scales of space and time. It may look something like this. And most importantly, we can examine it using both lower and higher order statistics. So here on the right, you see different level of statistics increasing from top to bottom. So the lower statistical properties would be univariate measures. Those could be the magnitude or the power. But most importantly, here we always focus on single regions. So this could be, for example, the activation or the level of activation within a single voxel, or as I just said, within the region. Then a little bit higher in the statistical order are bivariate measures. So here I mean functional connectivity, for example, seed-based connectivity, but most importantly, we only look at connectivity or interactions between two regions, only a pair of regions. And at the highest order of the statistics are the multivariate measures, where network analysis comes in. And the most important part is that we speak here about many regions and the interactions of many regions. As we know, the brain is really complex, and now we can see the brain as basically as a network. It makes sense to use those multivariate measures because we can not only determine single properties of the brain, but look at the topological large-scale organization and then see how little aspects may change those topology or the topological organization. To make this a little bit clearer, here you see what we can examine with those different kinds of statistical order. For example, with univariate measures, we can only examine a process, right? On, off, or on a continuous scale, like the level of the activation in a specific region. With the bivariate measures, we can examine interaction between two pairs, for example, two brain regions. But although the brain is built on a multitude of those little bivariate interactions, only higher order functioning as cognition or emotion are made possible by the combined interaction and the joint interaction of many of those little bivariate interactions in the brain, which make oscillatory patterns and therefore information flow throughout the entire system possible. And by using multivariate measures, we can actually examine this network in such a way. So here we look at patterns. And patterns can look like this. They can come in different kinds and flavors. For example, a dense pattern is really unlikely to exist in nature, or at least that the brain is organized in such a way, because obviously it requires a lot of energy consumption to maintain all those connections. In a dense network, it might be really efficient functionally wise because information can flow from one end to the network or from one node to another node on the really shortest path possible. But you don't see the true underlying backbone structure of this network, or at least here you cannot see which links are the strong links and which are the weak links, but you can see this by imposing something that we call sparsity on the network. So here in the bottom you see a sparse network, and there you immediately see the backbone structure of the network, which may hold the entire network together and allows information to flow from one node to another. At a fragmented network, it's obviously possible or not possible that the nodes from the outside communicate with the inside module because there is no way of going there, so you don't see any connections between them. In the connected network, everything is fully connected. I don't mean fully connected that all the nodes are connected, but every node can read information from one node can reach any other node in the network on a relatively long path, as you see here. And the problem is everything flows through this one major node in the middle. And it's obvious that if this node gets erased from the network, or we just remove it, then everything falls together. So it's really prone to disturbances if we make a target attack analysis on this specific red node here. An ordered network is kind of the same as a dense network, and as I said, it is really unlikely to exist in nature or in biological organisms because it's really costly. But what we have here is a really efficient way of parallel information transfer among the neighboring nodes. So neighboring nodes, because they're all connected and all neighbors with each other, they can take over information or information processing or kind of share the workload. In comparison to this, in the random network, we don't have this high clustering of the neighbors. So it is not really that any node can communicate with its neighboring nodes, which are in the vicinity. But the crucial aspect of those networks is that it's still, due to the random rewiring of the network, has a relatively short path length. So those random networks are characterized by a low clustering, but high efficiency by means of the shortest path. And how can we quantitatively assess those patterns? Well, we can use a branch of mathematics known as graph theory, and also a sub-branch when it comes here to neuroscience, complex network theory. And let's give you a little bit of an introduction about the history of graph theory, although it is now recently applied in neuroscience, and by recently I mean during the last 15 years, maybe. So in 2005, I think it all started. But graph theory is actually really older. So its founder is Leonhard Euler, who was born in 1700. And he lived in a city called Kaliningrad or Pregel. And he worked on a problem of crossing all the bridges that existed in this little city once and making like a tour, but not crossing any bridge twice, but going back to the origin again. And it's basically an optimization problem of how the shortest path of this route would be. And at the right, you see this formulation. And you can actually do this tour. It's called the Euler tour, and it's definitely on my bucket list as a graph theoretical neuroscientist. So I still have to do this. And this is a relatively simple problem, a simple system crossing all those bridges, but also more complex systems can be better understood when we describe them mathematically as graphs. So here at the left, you see a airport transportation cluster, and it looks actually really fuzzy, so we cannot see really what is going on. Maybe with our knowledge of where the continents are, we can think of where's Africa, America, and so on. We see there are some connections, but we really don't see how it is actually interacting. But by applying some really neat tools or magic of graph theory, we out of a sudden see, hey, there seem to be some different communities, some different networks, which within each of those communities have a really high density of links, so of connections among those airports, where there are just a few connections between those continents, between the clusters. And we also immediately see that there seems to be a difference in the importance of the different airports, as we see here by the size of the circles. And I say it again, recently neuroscience now adapted those graph theoretical methods for analysis of the brain. And as I said just now, by recently, I mean in 2005, so already 15 years ago. And why did we do this? We did it because it provides a really powerful way to quantitatively describe the topological organization of brain connectivity. As I said, by using higher order statistics in the form of multivariate analyses, we can examine patterns in the brain. So it gives us a way of transforming what we see here on the left, a really confusing mess, so to say, to a really neat organization where we can directly make inferences for the topological organization of this network. And here you already see a toy example of a graph, and that's it. A graph is nothing more. A graph is always composed of nodes and also of edges. So the edges are the little lines which connect the nodes, and the nodes obviously here are the dots, right? And I already put in some really basic property of such a graph, which is the degree of a node. And the degree of a node is equal to the number of its edges. And it already tells you something about how important a node is with respect to the topological organization of the network. So for example, this node with the degree of 0, it wouldn't matter if we remove it, so information could still flow from one side to another side in this network without being distracted. But if we would remove a node with a degree of 3, we already see that the organization or the structure of this network would change so dramatically that it may not be able to fulfill its actual function anymore. And this is just to show you that graphs can also look differently. So embedded networks are actually graphs. You'll see those probably in those nice scientific publications where people put a lot of effort in generating those figures. But when you work, like during work in progress, you'll probably not see those images too much, because then you will work with matrices, because matrices are equivalent to graphs. So graph or a network, here nodes are represented by the columns or the rows of the matrix, and the connection, so the edges between nodes, is represented by the respective matrix element. So that's only half of the story, because we could have two variants, one time the binary variant and one time the weighted variant. And for weighted networks here, those matrix elements, which I just said, those are continuous values, which can range from really strong, so high valued number, to really low numbered value, as you see in this little illustration. And for binary networks, those matrix elements are either 0, so they do not exist, or 1, they do exist. And just to finish with this little introduction into graph theory 101, it's really important for you to know that the graph, the embedded network, or the matrix are just equivalent visualizations of the same information. Okay, now let's come to complex network theory in neuroimaging, so the actual topic of this talk. And I'm going to read this out because this is written so nicely from Daniel Bassett. It's a modeling endeavor that provides a set of representational rules that can be used to describe the brain in terms of its subcomponents. So here we talk about the brain regions and their relationships to one another. So it could be the white fiber tracks or the functional connections, as I'll highlight in a couple of slides. So here you see that we can construct brain networks basically in two ways. One denotes the structural connectivity. So here we speak about structural networks. For example, you can do this by DTI, so diffusion tractography, by counting the streamlines, so the white fiber streamlines. Or we can construct functional networks, for example, fMRI, EEG, or MEG, which are constructed based on the large-scale temporal coherence of fluctuations of signals across time. And I want to establish now a little pipeline and to show you how we actually, what we have to do to derive at those brain networks. So in the beginning, we obviously start with some data. It could be, as I just said, fMRI data, DTI data, EEG or MEG data, and specific to the modality that you're using, you want to do some pre-processing that is appropriate. And right now I'm going to speak more about fMRI, specifically resting state data, because this is more my area of expertise. And here the problem is that it's not easy to choose the best pre-processing steps during the network modeling, because there is not a golden standard, at least yet, in my opinion, when screening the papers. And the problem why I'm telling you this is that network measures are very, very sensitive and will reflect more or less any change of network modeling in the network topology. This already starts with pre-processing. And only networks with the same modeling strategy should and actually also can be compared. And it is important to say this may not be too severe for simple pre-processing, but it could be more severe for the regression of covariates or the frequency range of the bandpass filter that you apply to your functional data. So just give you a little example. This is one paper from 2015 from Natasja Aurich. And there have been a lot of papers coming out that actually tried to evaluate the reliability of different pre-processing steps on graph theoretical models, measures in resting state data. And what you always see in those papers are those nice tables where they show a bunch of different pre-processing strategies, and I don't want to go into detail here, and they make comparisons and look how reliable, depending on the application of specific pre-processing strategies, the respective graph measures would still be. So what I would now suggest for you as a little bit as a take-home message, that a broad frequency range may result in more robust networks. Here, the little picture you see in the bottom is from another paper from 2015 from Martina Andenelli, and they could actually show that if you use other, so broader frequency ranges instead of only the standard frequency range that you would use for resting state connectivity analysis, which was established already back in the days of 0.01 to 0.08 hertz, then you have a higher retest reliability here measured by the intra-class correlation coefficient. And I would also suggest that you do not remove the global mean signal when you're working with those data. I mean, this is an always ongoing debate on whether to do it or whether not. So arguments for doing it is that it's correlation with respiration-induced fMRI signal fluctuations. So it's actually a nice noise, a noise canceling tool. But on the other hand, it shifts the distribution of correlation values of here you see in the bottom, this from a paper of Murphy and colleagues, the seed region in the negative direction. So it artificially centers it around 0. And the problem is, by definition, negative correlations would have to be discarded from the network for most of, for the, let's say, the traditional graph theoretic metrics. And this definitely would then affect the topology. So just leave it as it is, and then you're basically more on the safe side. And you also see here in this paper from Andinelli that the ICC is higher for at least some graph theoretic metrics here, the path length and alpha as well. And just to show you some more recent studies, the order of preprocessing steps may also impact the results. So this is a study from 2018. And even the choice of the motion correction pipeline may impact the results, this is a really nice paper from Lynn Parks and from Alex Fornido, based at Monash University in Melbourne. And okay, now that you've thought about how to pre-process your data and you've really chosen the best way according to your hypothesis possible, and according to the current literature which is out there, then you have to parcel the brain in different segments. Those could be the brain regions, as you see there. Then, and now I'm speaking more about functional construction of networks, extract some time series from those regions. Then you have to think of a way of how to combine those time series by means of a statistical coherence measure. Then you do this for all the regions, for all the pairs of the regions possible in your network, and you already have the graph in the end, and you can put this here as in kind of the association matrix. So this is called association matrix, where the row and the columns represent the nodes, and the matrix elements respectively represent the connection. So here the statistical coherence between the time signals that you extracted from your brain regions. Then you may want to apply some sparsity, so making it less dense, so throwing out some rather weak connections. And as I told you previously, some slides ago, this is already a network. So this is just a different visualization of our matrix, of our thresholded matrix. And on this, you can easily go ahead and compute those network topological measures. So basically do network analysis. And now I want to go through this pipeline and highlight a little bit more what to take care of during those different steps. And we'll start with the segmentation of the brain into different areas, the nodes, as we would say in graph theory. And well, it really depends on your research hypothesis, how you want to approach this. You may want to do a lot of reading, because I cannot give you the ground truth for your specific experiment. But there are different ways of doing this. So you could use pre-existing atlases. You could also use random parcellations. You could do it on a voxel-wise level. Obviously, then the network is really big, up to 20,000 voxels. You might want to use a supercomputer then for computing everything. Or you could, if you're also interested in gene interactions, you could use a psychoarchitectonic atlas, and so on and so forth. But there are only 3 rules which you would have to apply. So first, brain nodes should have intrinsic consistency. Then they should be extrinsic differentiation between those nodes. And obviously, they should be spatially constrained. And here's a little interaction with the pre-processing I want to just highlight for you. So you would have to mind the size of the smoothing kernel or even think of not applying smoothing, because if you smooth with a bigger kernel and then you segment the brain in non-overlapping areas, then already by application of the smoothing kernel, you are bringing signals from one region to another region, and then you're artificially increasing their connection. So this would not be good if you then, for example, examine the shortest path lengths, or yeah, in general, don't do it, I suggest, or think about how to do it. Then the next thing would be to think of how big the network should be. And this is an older study from Alex Fornito, Andrew Zaleski, and Ed Bulmer already, where they looked at network scaling effects. And what they show here on the graph, this is only for path lengths. If you apply a cost, so a density on the networks, which you would have to do, this is the sparsity constraint, which I talked before, which essentially means here at the cost of 25 that only 25% of all the edges possible in the graph would be maintained. Then it varies in the lower densities, so just 5% or 10%, which kind of parcellation you use. So here they had an ALL parcellation, and they go up to parcellation of around 1,000 nodes. But the higher the density is, so the more connected the network is, at least for the path lengths, it wouldn't matter anymore of how big your brain network is. So this is the network scaling effect that they describe here. Obviously, you would want to have a more granular focus on the brain if you are interested in more specific subregions. And obviously, if you just take the ALL atlas, 116 regions, including the cerebellum, then it wouldn't be enough if you want to study fine-grained differences in, let's say, cognitive functioning in subcomponents of the frontal cortex also. But in my experience, brain networks with a size of around 200 up to 300 or 360 nodes is a good compromise. Not having too much computing power, but also not to lose the granularity of the function of the different proper nodes of the brain network. Okay, if you've decided for this, then as I just said, you'll have to extract the time series signal and think of a way to combine those or to get a statistical coherence measure out of this. And what I meant is not only a coherence, but more or less a statistical dependence measure. So it could be a simple correlation between the extracted time series, could also be a partial correlation or mutual information. So this would, again, how to define the edges depend crucially on your research hypothesis and the question that you're interested in solving. So you can do this for resting state, then you'll simply take the time series from the beginning of the resting state scan to the end. But you can also do this for a task. So for an experiment where people actually do something, and you can try then to model or to include the task regressors. And for example, using tools like the CPPI toolbox, which was introduced, I think, in 2012 by Alex Fornito, which constructs task-dependent networks, or not only task-dependent, but condition-specific task-dependent networks. Obviously, you can also do this in the dynamic domain by not only taking the entire time series, but by partialing, segmenting the time series in little windows. So this is called sliding window approach, and only correlating the time series, which and the respective window with one another. Then you end up with different windows, which you can concatenate and look how network topological measures change over time. Of course, for the size of the window, you will have to keep in mind that you need a certain number of time points that the statistical dependence would actually make sense. So if you just have a window of, let's say, 7 time points, then I wouldn't trust so much in that the statistical dependence, let's say the correlation you examined, is really robust. So I think 40 seconds with the TR, so time of repetition of 2, so maybe 20 time points, is a good trade-off depending on how long you're scanning. And obviously you also have to think about the steps, so how you would slide the window across your entire time series. Important for you would still be to know that there can be an effect. For example, if there are 3 regions which are correlated with another, no, only 2 regions, so A and X and also X and B, directly highly correlated, then artificially you may also see that there is an indirect correlation of A and B So in the network, you would assume that there is a path between them, although they are not really connected. This could then be a problem if you want to examine the information flow from an additional region here C, from C to A. Then if you have this high correlation among A and X and B, then you would make the mistake of going from C to B to directly to A, and you would underestimate the path that the information would have to take actually over X. So that's why I do not prefer taking only simple correlation measures, because here you cannot guarantee that this effect is not present in your networks, but using rather partial correlations or regularized networks, for example, by lasso or L2 regularization, which you might want to look up, which is a really nice way of already introducing some sparsity in the network from the beginning. Okay, and then you're actually done with most of the part of network analysis, and then you come to the thresholding of the network. And I mean not with network analysis, but with the construction of the network. Sorry about this. Okay, what do you have to apply here? So here we are talking about building the graph. And here it's all about applying thresholds, so making it a little bit sparser, as I just said in the beginning of the talk. And here we have two possibilities. So we can apply an absolute or a proportional weight threshold. So the weight would mean the weight of the respective connections. And this will immediately solve the problem of really weak links, which tend to obscure the network topology of strong and insignificant or significant end connections. So basically the backbone structure of the network, which I talked about previously. So in the absolute thresholding, everything below a certain threshold is just set to 0, and in the proportional thresholding, a respective proportion of the highest correlations are considered, of the highest correlations are then considered as links. And we speak here of normalization by wiring cost, because you're normalizing it so that everyone, because assuming that everyone has the same number of nodes in the network, would then still have the same number of connections if you're applying this proportional threshold, which would make subsequent interpretation of your results actually easier and only feasible, to my opinion. So in the end, if we would have done this, we would have different kind of graphs. We could have binary undirected graphs. We could have binary directed graphs. So here the lower and the upper triangular of the matrix would code the information flow, the direction of information flow. Then we would have weighted undirected networks. This is what I use most of the time and what you will probably also use when using functional data. And if you have really advanced data sets, so that let's say effective data sets where you can make inference of causality from functional data, you could have also weighted directed networks. And obviously the load of information is higher or highest for those networks, here the weighted directed networks. But most of the time we are fine with analyzing weighted undirected networks. Obviously, again, there are some problems with edge definition, so with the thresholding. So because first of all, thresholding can definitely modify the network structure. So just to see this example, if you have a subject with a low connectivity and you threshold it by a fixed degree, k is the degree here, you'll have a gain of links. But with a high connectivity subject and you threshold it with the same degree, you'll have a loss of links. So you'll already here modify the network structure, and then you may lose the actual difference that you want to examine between those kind of individuals, because you're modifying the networks in a way that is too harsh. Then second, thresholding can also easily result in different stages of network fragmentation for each subject, and thus also a different number of nodes forming the network. So here, subject 1 has really high connectivity, and if you have a low absolute threshold, then everything is remained. But if you have a subject with a low connectivity, then links are already thrown out, and then you see immediately what happens. There are only 3 nodes kept in the network, and then you're comparing apples and oranges, because graph theoretic metrics on a network with 3 nodes are not the same as computing the same metric on a network with 5 nodes, right? And you also couldn't make inferences about the nodes which wouldn't have any connection to the rest of the network. So this would be a definite problem for you. Then There is another problem. So the low edge thresholds may also result in uniformity of the network. So, right, if it's just too low, the absolute threshold, then everyone would look the same. You couldn't detect any differences. And most importantly, results. So the association of the network topology with an independent variable. So let's say you want to examine age, and the association to the efficiency of the network may uniquely depend on the precise threshold value that you previously applied to each individual. One solution that actually covers all those problems that I just mentioned would be to construct each individual network over a range of thresholds by using this normalization by wiring cost, or the proportional thresholds. So starting by 10% of possible connections, increasing, for example, here in steps of 10% up to, let's say, 50%. And this then usually results in graphs, as you, for example, see here in a recent paper of Nestor and colleagues in addiction biology, where they compared cannabis-dependent subjects and controls. And here you see there's a significant difference in a couple of graph measures across this entire threshold range. And this is how, or what you want to see in the papers, so that people actually examine the association to an independent variable across varying thresholds, to make sure that it's not just an artifact on the specific threshold. All right, and now we can actually go and make some network topological analyses. Let's do it.